Here we're going to talk through some of the common mistakes and the mark scheme for the Unit 2 mock. As you can see, the grade boundary is out of 85. The grade boundary is very low. It's 66%, so 56 out of 85 for an A and 34 out of 85 for an E. It's quite a hard paper, which accounts for the low grade boundaries. This question is on classification and it was answered pretty poorly. If you ever get a diagram like this, I would put the hierarchy down the side. So your kingdom, phylum, class, order, family, genus, species. Like prawn curry or fresh green salad. So this tells us that the animals are the kingdom, the chordates is the phylum, the mammals is the class, the Perissodactyla is the order, the Equidae, etc. are the families, and then for the other things that we've got here, first name is the genus, second name is the species. So remember, that's the binomial method of naming things. Genus, then species. Genus with a capital letter, species with a lowercase letter, and it's in italics. First question is straightforward definition. Second question here, so 1A part 2, as you can see from the previous slide, there were three things in the family. There's the Equidae and the two others. The row next to the phylum was the chordata. Role of independent segregation. Now, something that a lot of my students missed out was putting the word genetic in there. You can't just put variation, it's genetic variation. And it does that really because you have the homologous chromosomes randomly align the equator. So you get a different combination of maternal and paternal chromosomes which moves into each daughter cell that's produced. This last question was an absolute stinker. Now, how to work out how to get about this. So you've got two different species and you should know that they're infertile. If they're different species, the offspring will be infertile, but the reason why in this case, so if you're having fertilization between the zebra and the donkey, you need to create gametes from both. Remember, gametes is produces haploid cells, one copy of each chromosome. So a gamete from a zebra would have 16 chromosomes. A gamete from a donkey, if you half 62, would be 31 chromosomes. 16 plus 31 gives you a total of 47. So the offspring from the zebra and the donkey would have 47 chromosomes. So a cell with 16 fusing with a cell with 31 to give 47. So the diploid number of 47, this is an odd number. Now that means that you can't form homologous pairs. There'll be one pair that's not got a partner. Okay, so you can't form homologous pairs and you can't split evenly 47 into two future gametes. Question 2, straightforward definition to start with 2A part 1, straightforward 2A part 2. This question on the whole wasn't too bad. You should notice the standard deviations immediately on this and use this question. Here we've got, so you just look for the highest number, so the highest number for that one, that's straightforward. If your standard deviations overlap, it means the difference is not significant. Now, this is a question that I thought was fairly straightforward, but people tended to make a bit of a hash of it. So, if you need to be aware that if a curve is to the right, whether that's through carbon dioxide shifting a curve to the right or from a different type of hemoglobin that's shifted to the right, you need to know that that lowers the affinity of hemoglobin for oxygen. You've got to say for oxygen. And it always means that you've got more oxygen unloading, so it can be supplied to your muscles or your tissues more readily. More oxygen can be supplied at a faster rate to your muscles. And why is that the case? Because you've got faster respiration, so you need more oxygen. Now, as you should be able to see here, we've got a large organism and you've just got a big round shape. There are no appendages like big ears, long tails, big feet. There's nothing to increase the surface area. So very, very tiny, small surface area to volume ratio. And that means that less heat is lost. I thought this question was pretty poorly, the question itself was pretty poor. You should be able to get the obvious describe mark. So you're describing, as you can see, the brain, same level before and after, but each of the other grey bars is less. The explain marks, so the brain's the same because it needs the same amount of oxygen. The lungs, obviously if it's underwater, the lungs aren't being used, there's less breathing going on because the seal can't breathe underwater, so there's less blood needs to go to the lungs. The heart rate's going to pump less because it's not got as much oxygen to get around the body. And the explanation for blood diverted to the muscles is because less is being taken to other organs, so more must be going to your muscles. And this is because the seal is swimming underwater. The idea then is if you've got less oxygen going to your heart, to your lungs, to your other organs, more is going to be conserved, so it can last underwater for longer. This question is quite a straightforward one. The key things are not saying it creates a steep concentration gradient, it maintains a concentration gradient, and it's across the whole length of the gill. Again, a straightforward question. This one was a little bit trickier. To answer this one, you first of all need to work out how much oxygen does the fish need. So you've given how much a fish needs per kilogram per hour. So the fish itself needs is only 0.4 kilograms. So the oxygen requirements of the fish is 0.4 times 90. Okay, so that's 40% of 90, which gives you 36 centimeters cubed. So that's how much oxygen the fish needs. Per decimeter cubed, you get 7 centimeters cubed. You get 7 centimeters cubed of oxygen. So you do 36 divided by 7. That means you need 5.14 decimeters cubed of oxygen to get enough out of the water. This question was fairly tricky. So the volume of water increases the temperature, it suggests why. So why does more water need to pass over the gills if the temperature increases? And it's actually nothing to do with increasing kinetic energy. So if the fish, because there's a bit of application here, but because the fish is cold-blooded, its rate of respiration inside it is dependent on its temperature around it. So everything that's not a mammal or a bird, if you increase the temperature, the rate of respiration increases. So that means it needs more oxygen. And it's a suggest question, so it is going to be off spec. The other reason is that there's going to be less oxygen dissolved in the water the higher the temperature. And you can actually see that in action if you leave a glass of water overnight. In the morning, when the water's warmed up, it's reached room temperature, you will see bubbles of oxygen on the glass. So that's water, so oxygen that's escaped out of the water, because as it's warmed up, less oxygen can be dissolved in the water. This question had tricky bits and fairly easy bits. These bits should have been quite straightforward. The biggest error I found was with this box here. If you're going to pass on a gene, it's got to be replicated first, otherwise you'd be passing it on but you wouldn't be keeping it yourself. That's no advantage. This was quite a straightforward mark as well. So if your ribosomes aren't going to be working, it's going to prevent protein synthesis. And how could that kill the bacterium? That means it wouldn't have enzymes. So enzymes for things like DNA replication, for respiration, enzymes that are fundamental for that bacterium surviving wouldn't be able to be made. This question was quite a tricky one as well. And it says that this question was really pretty tricky. So you've got to, the key is a single base was added. Now, a lot of my students thought that meant you had another triplet. No, it's a single base, so one letter is added. So trip here has been coded for, so different, first of all you can see straight away a different amino acid at this point has been coded for. ACC, that is different to ACG. There's been a C added at this point. So from that point onwards, you've had a frame shift. It's been pushed along one base. So where we had GGA here, we've now got GGA here, one base further along. And where we had CCA, sorry, start again. So we added a C here. So where we had GAA before, we've now got that GAA here. Where we had the CCA before, we've now got that CCA here. And you can see the CGA, we've got the CG, and actually the next base along will be an A. So we've had a frame shift, the base has moved one along in that direction. Cause a big change in a protein, a frame shift, because every amino acid after the mutation has been affected. The tertiary structure will be massively different to what it was before. So that's very different from just a substituting one base for another. So, added the code, and here we have got the letter. This is the base that's added. Standard mutation answer, usually it's related to an enzyme. So you've got different tertiary structure, so normally it's related to an enzyme that the active site changes shape, the substrate can no longer fit. This time we're talking about a channel protein, why the molecule can't pass through a channel protein. Pretty much the same logic, is that the molecule can't fit or pass through the protein channel. This question was very poorly answered, especially 6A. And it's using the immune system, so to work out how closely related things are. And it's all to do with the idea that if you mix antibodies, so if you mix, in this case, you're working out if humans are related to primates. So you mix human antibodies with the blood, the hemoglobin, from different primates. So this hemoglobin will contain antigens. And the antigens will be slightly different shapes. So the theory is the closer related they are, the more binding you have between your antibody and the antigens on your hemoglobin. That binding of the antigen and the antibody creates a precipitate. So the more precipitate that's formed shows that the protein structure is more similar, so they're more closely related. This is showing, you don't actually get any marks first of all for just stating this. So the largest decrease in temperature shows the most distant related. And why is that? It's to do with comparing the base sequences. So they've got fewer base sequences in common, so fewer complementary base pairs. So there are fewer hydrogen bonds. So that means that when you heat it up, the strands will separate at a lower temperature. Different species, organisms of the same species, might not separate at a really high temperature either. And that's because they've got different alleles. So the idea is you've got different sequences of bases on the two single strands. So you don't get 100% matching complementary base pairs. I thought this question was really tricky. So first of all, 20 million years ago was when the gorillas and the orangutans separated. They have a decrease in temperature of 3.5. So you need to work out what each 1 degree decrease is worth in million years. So you do 20, because it's 20 million years, divided by 3.5. So a decrease of 1 degree Celsius is 5.71 million years of evolution. So you then convert that to apply that to the situation between the humans and the chimp. In the table they have a value of 1.7, so there's a decrease in temperature of 1.7. So you do 1.7 times by the value per degree, and that gives you 9.71 million years. First part of this question is just knowledge, basic knowledge. You need to know these two things. These are the two things that you need to know for the index of biodiversity. Looking at this graph, first of all, you should definitely notice that as you're increasing the concentration of herbicide, we've got more crop yield and the index of diversity is decreasing. However, it does decrease after about 400 milligrams per decimetre cubed. Standard how science works question. So control allows you to prove that the results are due to whatever you're testing. So the results are due to the herbicide. This question here, so relationship between concentration of herbicide and the mean crop yield, but it's an explanation. So before we notice that increasing the concentration causes the crop to increase and then decrease slightly at higher levels. But why is that? So the crop yield's going to increase because more weeds are killed, so there's less competition. And it then decreases after the really high concentrations because the herbicide is damaging or killing the crops. It's toxic to the crops as well. This is your standard answer for reduction in the index of diversity. So if you're reducing the index of diversity, you're reducing the species diversity. If you're reducing the species diversity, you've got fewer habitats or niches and less variety of food sources. And not just less food, less variety of different food sources. So the idea that different animals can't be supported, or a large variety of animals can't be supported. There's not enough different types of food for different types of animals. So the standard long answer question for the structure of DNA, very similar to how you've got used to answering how structure of starch and glycogen and cellulose is related to its function. This was a question on DNA. So the idea you've got a double sugar phosphate backbone, so the bases are in the middle, so it protects the bases. A large molecule, so lots of genetic information can be stored. It's helical, so it's compact. The idea you can store a lot of DNA in a small space. The fact that it's made from a base sequence, and the base sequence allows certain amino acids to be placed in a protein in a certain order. It's double-stranded, that allows semi-conservative replication, so both strands can act as templates. You've got complementary base pairing, that means that during replication, identical strands of DNA are going to be made each time, except for a mutation. Weak hydrogen bonds, so you actually need to be able to unzip and access the bases, so you can break the hydrogen bonds, accessing the bases for replication. That also allows protein synthesis as well. And you've got many hydrogen bonds, so it's a stable molecule. This question was quite an easy 3 marks as well. I bet a lot of people put an answer and thought there's no way I'm getting 3 marks out of this. So all you need to say is E, so you've got mutation in E, produces the highest risk. Mutation in D produces the next highest risk, and a mutation in C produces the least highest risk. This graph, just looking at it, and we'll do the describe question first, which is over the next slide, comparing the 2 lines, and then we'll move on. So you should notice that as time increases, so as time's going along, here is when you're adding the drug. So every time you add the drug, both lines decrease, level off, and then increase. You can see this here as well. Decrease, level off, then increase. A lot of people, when describing a similarity, forgot the level off bit in between. You can see every single time on all of them you level off. Also, you can see that they level off for the same length of time. So it's about 4 days. Each time it's about 4 days that it levels off for. So they level off for the same length of time. The difference is that the healthy cells decrease faster. Okay, so healthy cells decrease faster, but they also increase quicker. So they're the 2 differences. I could not believe how poorly people answered this question. So rate is the change divided by the time taken. always, the examiner is expecting to see you annotating your graph. So with a ruler, I've gone up and across. So from day 42, so the difference, so you've got 10 bars, which is worth 20. So each little bar is going to be worth 2. Okay, so I've gone across 1 bar for 42, up and across. I've gone across 3 bars for 46, go up and across. A lot of people misread this scale as well. You've got 5 bars, which is worth 200. So each little bar is worth 40. So this one here is 280, and this one here is 560. So the difference in the number of cells is 1280 minus 560 to give you 720. The difference in time, so the time taken, is between 46 and 42, which is 4 days. 720 divided by 4 gives you 180 cells killed per volume of blood per day. There's absolutely no room for reading incorrectly. They could have asked a question, they didn't, but why is an advantage to do per unit volume of blood? And it's the idea that different people have different volumes of blood, so it allows comparison. So here we go, that's the mark scheme for the question that I went through. This one here, suggest question again, so it's going to be off spec. You've got to think outside the box. They unusually make this, they don't usually make it more than 2 marks. This is a 2 marker. So I did, and this was quite an okay one to get. If you're going to give more of the drug or more frequently, too many healthy cells are going to be killed. It could actually make somebody die or have really serious side effects, make them feel very, very, very ill. And it also takes time to replace the dead cells, so to create enough healthy cells so the person doesn't feel too ill for too long. This question was pretty tricky. Right, with this question, I'm just going to show you a couple of things that you should have picked out from the text. So first of all, nicotine, when it reaches the brain, this bit here, it binds to a specific protein. So when that happens, you get reward to the smoker, and that's why people find it difficult to stop smoking. Got a large number of volunteers, very few people picked that up. And it's once a month for 5 months. The experiment only lasts 5 months, and you do have a placebo, so you do have a control. So, double-blind trial, standard answer. So you need to know that. Suggest again, so we've had a few suggest questions. Remember, it's a list draw, so it says 2 reasons, so don't ever write more than the number it says. And here are some of the things that you could have had. So all things that that could have affected the amount of nicotine in the blood. So for this question here, so the vaccine, you need to know that with the vaccine, it does use information from Unit 1. You've got to get used to that your knowledge is cumulative, you can't forget anything. So your vaccine would mean that you produce memory cells. So if you smoke, it will trigger the memory cells to produce plasma cells, which produce antibodies. So that's the linking to this first mark. So the antibodies, using the antigen binding site, will bind to the nicotine. If it's bound to the nicotine, it's going to prevent the nicotine from binding to the protein in the brain. If the nicotine can't bind to the protein in the brain, then you don't get the reward for smoking. It doesn't satisfy your addiction. So there's no reason you don't get the reward, you don't want another one. So some people have suggested that vaccines should not be given to smokers. So I've split this into agree and disagree because we've got an evaluate. So it's always agree and disagree, or for and against, or pro and not pro and against for an evaluate. So reasons you might agree. So people knew the risk before they smoked, so kind of the idea that it shouldn't be given any sympathy. And really important point here, should the money be better spent elsewhere treating other health problems that maybe aren't smoking inflicted problems? However, NHS, it's unethical not to treat people. Okay, people could still die, it's unethical not to treat them. And it could be the idea that maybe more research is needed, but it could mean that less money is needed in the future to treat the diseases that are caused by smoking or cancer. Because it does require a lot of money to do that. The NHS uses a lot of its budget to treat smoking-related diseases. We're going to continue. Looking at this graph, you should notice that all the antibody shapes are the same. There's a much bigger response in the high antibody responders, but all the lines, apart from placebo, increase up to 5 and then decrease. So after 5 months, the antibody concentration is decreasing. So only a month after giving the vaccine, the antibody concentration does start to decrease. So let's look at this table. So looking at this table, so second piece of evidence from the experiment, high antibody responders. So this means that more people than not, so it's got over 50%, so more people than not stop smoking. With all the others though, okay, with you've got less than a third with the low antibody responders, and there's not much difference really between those two. So the placebo, even with the placebo, nearly a third of people stop smoking. So let's look at what we should have. Now this question, does the data support this statement? That's leading you to another evaluate. If you ever get that, it's meaning you want for and against. So explain, does it support? So I'll split it into whether piece of evidence that think support the idea it's a major breakthrough, and piece of evidence that kind of issues with the breakthrough. Maybe it's not such a big breakthrough. So first of all, the idea that the high antibody responders are more likely to stop smoking. So we saw that number was over 50%. Another point 4 is the placebo had the lowest percent to stop smoking. So the idea that all the antibody ones are higher than the placebo, have a higher chance to stop smoking. And it did tell us in the text we've got a large sample size. So always put these both together. This means it's representative and reliable. Against though, okay, so against this being a major breakthrough. So it does not tell us anywhere how many of that large sample size were high antibody responders. It might have only been a couple. Okay, so we've got lacking a piece of information there. One or the other for these two here. The fact that the low antibody responders had a similar response to the placebo. So there's not much difference if you're a low antibody responder. The antibody levels drop after 5 months, so you'd need boosters. Okay, so you know, is the vaccine actually that successful if you need a booster every 5 months or every month? It doesn't tell us any information at all in the table above if people started smoking again after 6 months. Okay, is it a long-term solution? We do not know. And again, we've got a risk factor that's being so causation, correlation type question. So nicotine may not be the only factor making people smoke. There may be other reasons why people smoke. So psychological factors, stress, other things will be affecting whether people smoke.